challenge. If you can solve this challenge, it would open many new avenues. Right? So that's why enzyme immobilization ma pani there's a lots of interest किन छ भन्दाखेरि why there are so many interests industrial application haru huncha, right? Different applications haru huncha, food ma, diagnostics ma, industry ma. That's why it's one of the major area of research currently. One of the major current area of research. So if we want to cellular systems enzymes industry it's always good if the enzyme is immobilized. Solution ma hunu enzyme free state ma hunu, right? Or the solution state ma hunu, or it has been fixed to a stationary support. That's a very difference. The cost ma, effectiveness ma, efficiency ma, overall the process flow, efficiency, effectiveness, cost, economy, एकदमै reduced गरिदिन्छ drastically in the case of an immobilized enzyme. That's why many publications and patents are published each year. That is on enzyme immobilization methods, techniques, and applications. So even though we have come a long way, right, from 1916 to 2021, it is still an active area of research, right? So many published research articles and review papers are being published. So many enzymes, right? So such as penicillin G, acylase, invertase, lipase, and proteases are being integrated in various large-scale processes, right? So like these are some of the major enzymes, right? So penicillin G, acylase, one of the major important, right? The penicillin, one of the major antibiotics. What is the major antibiotics still, right? These are these penicillin, right? Which contain the beta-lactam ring. That is a characteristic beta-lactam ring. That beta-lactam ring is industrial process. There is an industrial process to synthesize the penicillin. The industrial process it can be little. Industrial process there are always some limitations. The limitations are like remove certain steps or the chemical steps non-enzymatic enzymatic steps substitute this major enzyme penicillin G acylase enzyme. Industrial enzyme, one of the most widely used industrial enzyme. Industrial enzyme penicillin or the antibiotics sector use the major enzyme. So there's always this research interest, right, to immobilize such enzymes that have that's major industrial importance. So actually, enzyme can be attached to almost any solid support that is inert, organic, inorganic, or the insoluble material. So one of the major focus is that the enzyme shouldn't chemically react with the solid support, right? It should be inert, right? That's one of the major consideration. So enzyme mobilization has several advantages: higher stability, right? Higher stability of the enzyme, more reusability, longer storage time, a broader range of activities in presence of various physical and chemical factors, and easier product recovery. So enzyme mobilization ko धेरै advantages. That's why we are opting for the enzyme immobilization. So it's very difficult. It's very difficult to immobilize enzyme and retain its enzyme activity. So that's why this is a major problem. The technology of the immobilized enzymes have evolved significantly during the past 100 years, but still it is known as a technique of trial and error, as there are many attached queries that remains unanswered. That's why this is we still use like trial and error techniques, and that shows what is our level of understanding about these enzymes. We know so little about enzymes. This nano biocatalyst, right? This organic biocatalyst, we know so little about them. That's why it is very difficult for these enzymes to be immobilized. हामीलाई थोरै कुरा थाहा पाएको कारणले एन्जाइमको स्ट्रक्चरको बारेमा, प्रपर्टिजहरुको बारेमा, यसको सेप र स्ट्रक्चर र फङ्सनको इन्टररिलेसनसिपको बारेमा we know very little about enzymes है त हामीलाई धेरै थाहा पाए जस्तो लाग्छ we still have very little understanding. That's why this science is still reliant on the trial and error techniques, right? So there is no field where immobilized enzymes do not have an application, right? We'll study here, right, about many different applications of the immobilized enzymes. So the recent advancement in the nanotechnology has revolutionized enzyme immobilization due to the generation of the large number of nanomaterials. So now our enzyme immobilization, it is getting fused with the nanotechnology, right? There are new nanomaterials, structural science right? The material science where so there are new materials that are being discovered, and these new materials, they'll act as a stationary support upon which our enzymes, they will be immobilized, right? So it's an exciting time, right? Exciting time to study about the immobilization of these enzymes, right? Exactly, so like everybody will say this is the best time to study. I think enzymes study ko barema chahi this is the best time, right? It's a time where lots and lots of research have been carried out. एकदमै interest छ, right? There's a industry को लागि, research को लागि, र government को लागि, everywhere चाहिँ they are एकदमै momentum चाहिँ carry on हुँदैछ, right? Around this immobilization of the enzymes or nanomaterials हरुमा पनि. So let's talk about history, right? So that we can have some basic idea. Research के भएको थियो and past ma, and what were the research that were carried out, and that's why we can move forward, right? So 1916 to 1940 is called the early phase where the various immobilizing matrices were discovered. So predominantly belonging majority to the simple organic carriers such as glass, ammonia, hydrophobic compound, coated glass, etc. for the attachment of the enzymes as well as other biomolecules like immunoglobulins, organelles, or the competent cells. So there were many. So at least then, like 1916 to 1940s, it was an era of discovery. It was the era of random discovery, right? So we had all these kinds of different materials in the lab, right? It could be any material in the lab, and the scientists, they try to immobilize those enzymes upon those solid material, right? Still, still science, right? The biological science is still biological science, material science in general is still reliant on trial and error, right? We have too little understanding of the fundamental concepts को बारेमा नै हामीले अझसम्म थोरै understanding भएको छ. Interactions at the nano level, है त त्यो angstrom levelमा, nano levelमा हुने interactions को बारेमा we know so little. That's why still the science is reliant on the trial and error, right? अब यसमा चाहिँ के गरिन्छ भन्दाखेरि ल्याबमा के छ त, what are the different materials in the lab, right? Any different kind of materials, and what you do is that you try to immobilize those cells, those enzymes, right, upon those solid material, right? So all immobilization methods were used, were reversible in nature, particularly based on adsorption. So what were the techniques? What were the interaction? Interaction say it was reversible, so it was based on the adsorption, right? Adsorption, right? Adsorption is a surface phenomenon, right? You should be quite clear with the word adsorption. Molecular level, my adsorption meaning ke huncha bhanera hai ta. This is the word something you will encounter very often, right? So actually adsorption is a surface phenomena huncha. Surface ma chai ke huncha, these molecules, right? Enzymes, or it could be any molecules, right? It can be gases or liquid, right? Abisma chai ke huncha, they adhere, right? They adhere to the surface, right? The solid surface or the surface. Actually, film banaera adhere. So there are some chemical interactions. There are some interactions, and normally these interactions are reversible interaction, covalent interaction हुँदैन, maybe hydrogen bonding or the van der Waals or the dipole-dipole interactions. These are the different kinds of interaction that are predominant in a reversible interactions that are mostly this adsorption phenomenon, right? Okay, right. Similarly, so from 1940s to 1965 onwards, right, this is the underdeveloped phase, and this phase saw the transition of the immobilization method from the physical adsorption, right, to the specific ionic adsorption, having more specificity and durability. So early phases में के थियो, the interactions were quite reversible, है त? But more researchहरूले के देखायो भन्दाखेरि that the physical adsorption like we could move forward, right? There is this small development, right? There is uh, some breakthrough in this immobilization techniques. So there were these techniques like these ionic adsorptions, they are more specific and durable. So various matrices like phosphocellulose, DEA cellulose, right? Di amino ethyl cellulose, amberlite XC97, Dow X2, right? These are these ion exchange reagents, right? You, these are proprietary names, right? These are proprietary names, but what you should understand is that they are basically these are ion exchange reagents. So as well as the additives, right? Synthetic polymers, polyamine styrene and polyisocyanate, and inorganic materials like carbon, glass, kaolite, and clays were used. So this was an era of interesting discoveries. I think there were new materials, synthesized materials, they were being discovered. So in the developing phase from 1965 to 1970, this phase mainly focused on enzyme immobilization by covalent method using various chemical cross-linkers. So one of the major cross-linkers was the butyraldehyde was introduced for enzyme immobilization for enzyme attachment onto insoluble carrier, natural and synthetic carrier. So there was this cross-linker concept arrived, right? So rather than directly attaching the enzyme to the solid support, right, the scientists, they had the idea of developing these chemical cross-linkers. These cross-linkers, they would join the solid support to the enzyme, right? So one of the first cross-linker that was developed, that was introduced, that was synthesized was our butyraldehyde. So synthetic carriers were also developed with active functional groups such as polyanhydride, polyisothiocyanide, etc., which were used for enzyme attachment directly to the support, right? So there are these synthetic carriers. They have these functional groups like polyanhydride, polyisothiocyanide. So that helps them to attach directly to the support. Many carriers of different physical or chemical nature, different hydrophobicity and hydrophobicity or different shapes or sizes, be it cis, film material or capsule, were developed in order to provide carriers with sufficient diversity, right? Or lots and lots of material of these carrier molecules were developed, right? With different chemical nature, right? Different hydrophobicity, different shape and size, and were developed between 1965 and 1970. And from 1970 to 1980 onwards, so this phase included exploration of novel matrices for enzyme immobilization using established basic methods, including covalent adsorption, entrapment, and encapsulation methods, right? So these methods, it was the 1970 and the 1980 was the culmination of all the efforts, right, of all the techniques that were developed earlier. So this is always, that's why we're studying about the history, right? How these historical development they occurred, right? And what knowledge, right? What wisdom they had to offer. Various other new immobilization methods such as affinity binding and coordination binding were developed that had industrial potential. right? So नयाँ methods like affinity binding and coordination binding were also developed in this era. Even basic methods were modified in order to increase their efficacy. For example, entrapment of the enzyme in the gel matrix by cross-linking using covalent or affinity adsorption in order to avoid leakage. So let us study about the entrapment. So entrapment पर्दाखेरि one of the major problems चाहिँ के छ भन्दाखेरि enzyme leakage हुने, right? I think you have already studied. That's why entrap गरेको छ, right? एउटा कुनै matrix materialमा, right? Gel materialमा. So gel materialमा चाहिँ के हुन्छ, there is always the chance of leakage हुन सक्छ. To leakage के गर्न सक्छौँ भन्दाखेरि we can avoid that, right, by cross linking using covalent or affinity adsorption, right? So these enzymes, they can be retained, right, with higher efficacies, with higher probabilities. So various new chemical reactions were identified for enzyme coupling by covalent methods to carriers. So नयाँ chemical methods, right, नयाँ linkage methods they were developed. The most important achievement was the uses of immobilized penicillin G acylase, the production of 6-APA, that is the intermediate in the synthesis of beta-lactam antibiotics, and the production of fructose syrup by using immobilized glucose isomerase. So 70s and 90s, it showed great promise, right? Their promises are dekhaayo, and even the industrial production was achieved with the help of the immobilized penicillin G acylase. Right? One of the major, right? One of the major enzyme, penicillin G acylase. And this all enzymes like glucoisomerase that is used in the fructose syrup production. There was some development. So enzyme immobilization in solution by catalyst behave as any other solute in that they are readily dispersed in solution or solvent and have complete freedom of movement in the solution. So aqueous medium enzyme राखियो भने के हुन्छ, it can move in all the direction. It's all diffusion, right? The laws of diffusion control its movement with a more or less complete freedom हुन्छ, like random walk भन्छ नि. So the enzyme को path कस्तो हुन्छ भन्दाखेरि the random walk जस्तो हुन्छ, and so त्यसको कारणले के हुन्छ भन्दाखेरि let's limit the degree of the freedom of the biocatalyst. अब एकातिर के छ भन्दाखेरि there is substrate molecules and we have the enzyme molecules, and even substrate, they have their own degree of freedom, right? They are allowed to move freely in the aqueous medium, and even our enzyme, it is allowed to move freely in the aqueous medium. So what it creates is that so chemical reaction they have to bump together, right? They have to bind together. So के हुन्छ भन्दाखेरि that if both are independent, independently move गरिरहेको छ भने it becomes difficult. So हाम्रो technique के छ भन्दाखेरि we want to immobilize the enzyme and limit its degree of freedom. So immobilization separates a biocatalyst from the bulk solution, right, or the aqueous solution phase to produce a heterogeneous two-phase mixture. Right now we want to create a two-phase mixture. एउटामा चाहिँ we want to create the immobilization enzyme, अर्कोमा चाहिँ we want to create that the substrate वाला medium. Like two-phase mixture बनाउन हामीलाई चाहिँ we'll try. And there is our soluble enzyme plus substrate level, it gives product single time uses, or immobilized enzyme plus substrate gives product, but you can use the enzyme again and again. So normally के हुन्छ भन्दाखेरि product recovery गर्दाखेरि चाहिँ it's better, right, to throw away the enzymes. Product recovery एकदमै गाह्रो हुन्छ कि otherwise if you are trying to recover the enzyme recovery becomes very difficult. That's why the term immobilized enzyme refers to enzyme physically confined or located in a certain defined region space with retention of the catalytic activities, and which can be used repeatedly and continuously. This is how we like to define the immobilized enzymes. So what are some of the advantages of enzyme immobilization? So protection from degradation and deactivation, right? While you are immobilizing the enzyme to a solid support, के गर्न सकिन्छ भन्दाखेरि त्यसको चाहिँ half life you can increase its half life because you are attaching it to the solid substrate. You can make it more stable. The deactivation you can prevent the different proteases or different factors or let's say enzymes they can degrade or deactivate. And while you are trying to immobilize those enzymes, so you can protect, right, from the deactivation and degradation. So retention of enzyme and the enzyme free product. Since the enzymes are retained, our final product will always be free from enzyme. So cost, post processing cost it becomes very economical, right? Because your enzymes they are not as a contaminants. Normally enzymes are contaminants ko rupma chai huncha. That's why post our recovery of our metabolite becomes easy. So it is recycling and repetitive use. So cost efficiency, enhanced stability of those enzymes. So use as a control release agent, right? So you can use these enzymes, right, whenever you want, right? You can remove them. You can do whatever you like. So this is your material immobilized by currently you can remove them, right? So control release agents they can be used. Ability to stop the reaction rapidly by remove the enzyme from the reaction solution or vice versa, right? You can add, you can remove, it can be done very quickly. Also allows of the multi-enzyme reaction system. Multi-enzyme reaction system you can develop, right? Series of reactions every substrate every material what you can do is you can embed those enzymes. A series of reactions you can carry out. So lots of there are many advantages, right? That's why industry at the university there is so much excitement about enzyme immobilization. So one of the most important if you are studying about the enzyme immobilization, you should study about the carrier matrix, right? The solid support. The matrix of the solid support immobilizes the enzyme by holding it permanently or temporarily for a brief period of time. So there are a wide variety of matrices or carriers or support available for immobilization. The matrix should be cheap and easily available, right? It should be economical. Industrial application this is always a major criterion with lab ma काम गर्नको लागि, right? Analyte separate गर्नको लागि maybe it's not required. Diagnostic purpose maybe it's not required. But if you want enzymes to be used in industries, the matrix should be cheap and easily available, inert universal, right? There are many characteristics. So what are the ideal characteristics? This should be economical, inert universal. It shouldn't react with substrate. It shouldn't react with enzyme, right? Physical strength, stability, ability to enhance enzyme specificity, activity, regenerability, ability to reduce product inhibition, ability to prevent non-specific adsorption and bacterial contamination. So all these are some of the properties of an ideal carrier matrix. So there are different kinds of carrier matrix. So for example, so there can be natural polymers, right? They are alginate, right? And these are natural polymers. They are derived from the cell wall of some algae, right? And calcium or magnesium alginate is the most commonly used matrix. They are inert and have a good water holding capacity. So even I think in your lab you'll use this alginate, quite economical. Cost it is very lab scale and industrial scale alginate is usually preferred, right? Alginate. So chitosan and chitin, they are structural polysaccharides occurring naturally in the cell wall of fungi and exoskeleton of the arthropods. Various functional groups in the enzymes can bind to the OH group of the chitin and can form covalent bonds. Similarly, there is this collagen, right? And there is this carrageenan, that's sulfated polysaccharide obtained from some red algae. The group's gelling properties together with the high protein now holding capacity makes it good support for the immobilized enzymes. There are this gelatin, cellulose, starch, and pectin. So all these they can be good support material for the enzyme immobilization. So there are the synthetic and the inorganic carrier material, right? Synthetic, they good ion exchange reagent or polymers, and they are insoluble support with a porous surface. So their porous surface can trap and hold enzymes or the whole cells. For example, diethyl amino ethyl cellulose, right? DEA cellulose, polyvinyl chloride, PVC, UV activated polyethylene glycol. So these are some of the synthetic material that can be used as the immobilized medium. Of inorganic material, for example, zeolites, microporous aluminosilicate minerals with good absorbing properties are extensively used for immobilizing enzymes and whole cells. Ceramic sorugma, right? Diatomaceous earth, right? Silica, glass, activated carbon, and charcoal you can immobilize, right? So now we'll study about the methods of enzyme immobilization. So there are different methods of enzyme immobilization. There are these physical methods and chemical methods. In physical methods, we study about entrapment, adsorption, and microencapsulation. And in chemical method, we study about the covalent attachment, cross-linking, ionic binding, and conjugation by affinity ligands. So entrapment, as the word describes, there is this polymer, and that is going to entrap the enzyme, right? So this involves cross-linking of the enzyme to the polymer, right? You can imagine the polymer as a spherical surface or a spherical ball, and inside the spherical ball, there is our enzymes. Enzymes ready to catalyze the certain chemical reaction. So in every direction, covering almost every side chain present on the surface of the enzyme by physically entrapment within a polymer lattice. So there is this polymer lattice, and inside the polymer lattice, you are going to entrap these soluble enzymes, right? That's why we are studying about the problem of leakage. There is always that probability. So it allows permeation of appropriately sized substrate and release of molecules, which ensures continuous transformation. However, this method can only be used in limited number of enzymes. And key that there is this polymer lattice, and this polymer lattice will allow the entry and exit of the substrate and the product material, respectively. So there is this continual transformation. The substrate, they can freely enter inside the polymer lattice, and after the conversion, the substrate is converted to the product. The product, they are released, right? They can exit the polymer lattice. This method has several advantages: simplicity, no change in the intrinsic enzyme properties, right? You are not modifying the enzyme in no way, right? You are using enzymes as they are. You are just enclosing them or entrapping them in a polymer lattice. So it involves no chemical modification, minimal enzyme requirement, and metrics are available in various steps. Various disadvantages of these methods are enzyme leakage. Only small size substrate products can be used. It requires delicate balance between mechanical properties of the matrix and its effect on enzyme activity and presence of diffusional constraints. So these are some of the disadvantages that these entrapment techniques poses. So what are the methods of entrapment? That is the inclusion in the gels. The enzymes are trapped inside the gels, or the inclusion in fibers. That is the enzyme supported on fibers made of the matrix material. And inclusion in microcapsules. Enzyme entrapped in microcapsule formed by monomer mixtures such as polyamine and calcium alginate. So these are different methods by which you can entrap these enzymes. So what are some of the advantages and disadvantages? Advantage of entrapment, fast method of immobilization, right? So there is nothing much happening, right? It's quite quick, it's cheap, low cost matrices are available, right? So mostly used whenever you say calcium alginate, with the calcium alginate are most commonly used. So easy to practice at a small scale. Mild conditions are required, less chance of conformational changes in enzyme. Why? Because you are not doing any chemical modifications. You are just entrapping the enzymes, and it can be used for sensing applications. Sensing applications or normally these can be used. What are some of the disadvantages of entrapment? These are the leakage of enzymes, poor diffusion limitation, chance of microbial contamination, and not much success in the industrial process. So these are some of the disadvantages. So next is our adsorption. So Nielsen and Griffith used charcoal to adsorb invertase for the first time in 1916. Enzyme is attached to the support material by non-covalent linkages, including ionic or hydrophobic interactions, hydrogen bonding, and van der Waals forces without any pre-activation of the support. So enzyme actually enzyme is attached to the surface of the support material, and there are mostly checked by the non-covalent, right? The non-covalent link is that's why this is a reversible process. So what are some of the common interactions that occurs adsorption? These are ionic or hydrophobic interaction, hydrogen bonding, and van der Waals forces. The dipole-dipole interaction. So the matrices used are either organic or inorganic in nature. For example, ceramic, alumina, activated carbon, kaolinite, bentonite, porous glass, ketosand, dextran, gelatin, cellulose, and starch. These are some of the commonly used adsorption metrics. So immobilization method involves optimization of variables including pH, temperature, nature of the solvent, ionic strength, concentration of the enzyme, and adsorbent. So these are certain parameters or different variables that you need to optimize with, because these forces, right? These non-covalent linkages, the strength of these forces depend upon these quantities, right? These parameters. So here the enzyme is directly added to the surface active adsorbent without the removal of any adsorbed enzyme during washing. This method is simple and mild with vast variety of carriers, helpful for simultaneous purification as well as enzyme immobilization. Example would be asparaginase on CM cellulase, carboxymethyl cellulase without any conformational change. However, it involves intensive optimization due to the involvement of a number of factors that play a role in enzyme deabsorption following slight changes in its microenvironment. So that is pH, temperature, solvent, ionic strength, and a high substrate concentration. So it's very simple method, but there is this intense optimization that has to be carried out. Right? Because microenvironment, or say if there are slight changes, then let's say enzyme deabsorption, it can lead to the deabsorption. So what are some of the methods of adsorption? Some of the methods of are static process, where the immobilization to carrier by allowing the solution containing enzyme to contact the carrier without stirring, right? It's my static process, so you don't need to stir. So these enzymes, they can come into the contact of the solid material without stirring, right? So that's the static process. In the dynamic batch process, the carrier is placed in the enzyme solution and mixed by stirring or agitation. So dynamic batch process, you need to apply certain external force, right? That is the stirring or the agitation. And there is this reactor loading process. The carrier is placed in the reactor and then enzyme solution is transferred to the reactor with the continuous agitation. And there is this electrode position process where the carrier is placed near an electrode in enzyme bath, and then the current is put on under the electric field. The enzyme migrates to the carrier and deposited on its surface. Right? There are so different techniques, right? Adsorption there are different techniques. So what are the advantages and disadvantages? Advantages are no pore diffusion limitation, right? Since this is a surface phenomenon, right? The pore, you don't require any pore materials, right? And it is easy to carry out. No reagents are required, right? So there was no any chemical transformation that was occurring. With the enzymes, they were being linked with the substrate material. There was no any chemical or let's say the covalent interaction involved. Covalent interaction, let's say, we need to we have to be extra careful because it can lead to the conformational change of the enzyme. Enzyme conformational change, say the behavior structure. If there is a covalent interaction, if there is non-covalent interactions, right? So no external reagents are required. So minimum activation steps involved. Comparatively cheap method of immobilization, less disruptive to enzyme than the chemical method. So what are some of the disadvantages? So disadvantage is that deabsorption चाहिँ हुन सक्छ है त. Absorption पनि हुन सक्छ, deabsorption पनि हुन सक्छ. Deabsorption भयो भने the enzymes they will lead to the solution. Solution मा आइहाल्छ. So efficiency पनि quite less है त. किन deabsorption हुने process चाहिँ हुन सक्छ कि micro environmentsहरुमा change हुन सक्छ. That is very difficult to control, right? Overall solution को pH चाहिँ you can regulate. But एकदमै सानो पोरहरुमा सानो thin film मा हुने pH चाहिँ it is very difficult to control. That's why there is this always the chance of deabsorption from the carrier material. That's why the efficiency is quite less. So another method is the micro encapsulation. So enzymes are immobilized by enclosing them within a spherical semi-permeable polymer membrane with a controlled porosity, right? So these are quite small. That's why these are called microencapsulation. So the porosity can be controlled and it is usually limited between 1 to 100 micrometer. Semi-permeable membranes can either be permanent or non-permanent membranes based on the constituents. Constituents basis these membranes can be permanent or non-permanent. So the permanent membranes are made from cellulose nitrate and polystyrene, while non-permanent membranes are made from liquid surfactant, right? So these membranes are also used in encapsulation of dyes, drugs, and other chemicals. So enzyme immobilization by encapsulation have extremely large surface areas due to which they have greater catalytic efficiency, right? These are since they are very small भएपछि के हुन्छ? So volume कम हुन्छ, volume कम हुने बित्तिकै ratio of volume to ratio of surface area चाहिँ के हुन्छ यिनीहरुको हाई हुन्छ surface area धेरै हुन्छ है त surface area धेरै हुन्छ volume थोरै हुन्छ है त that's why they have higher catalytic efficiencies compared to the encapsulation method. However, there are several reports of occasional inactivation of the enzyme and despite presence of high enzyme concentration. So even there are these certain inefficiencies that can creep into. So the next is the covalent methods. Covalent method, but there is this certain covalent bond that is formed between the solid material and the enzyme. There can be this there can be this linker molecule so the enzyme is attached to the matrix by means of covalent bonds. And what are the different covalent bonds? Covalent bonds are the diazotation, amino bond, SNP base formation, amidation reaction, thiosulfide bonds, peptide bonds, and alkylation reactions. Different chemical bonds. So the enzyme molecules are attached directly to the reactive group, right? They can be this about the hydroxyl group, right? The amide group, the amino group, the carboxyl group, present on the matrix or by the spacer arm, right? Which is artificially attached to the matrix through various chemical reaction, right? Spacer arms or spacer linker or these are the similar, right? Diazotization or the SNP base or the imine bond formation. These are some of the common covalent bond that are formed. Right? So matrices commonly used are either natural, glass, spherics, agarose, or cephalose, or synthetically manufactured such as acrylamide, metacrylic acid, or the styrene. Right? So both kind of materials can be used. The selection of a particular matrix depends upon its cost, right? Cost, availability, binding capacity, hydrophobicity, structural rigidity, and durability during various applications. So always, right, the different factors are like we have to consider. So method of immobilization involves non-essential amino acids other than the active site group leading to the minimal conformational change. So every time while you are making this covalent modification, these covalent modifications they have to be made on the non-essential amino acids. To structure structures of scaffolding regions, the regions that are not very important, right, other than the active site groups use because that so that we can minimal conformational changes house so that enzyme activity might the drastic change now. So it helps to promote higher resistance of immobilized enzymes towards extreme physical and chemical conditions. Right, so minimal conformational changes covalent interactions this can be resistance to temperature, denaturants, or organic solvents. However, this method of immobilization leads to greater strain on the enzymes and sometimes lead to the drastic changes in the conformation and the catalytic properties of the enzyme due to harsh immobilization condition and occurrence of similar amino groups at active site being involved in interaction of the enzyme with the matrix. Disadvantage part okay, covalent modification introduced you will attach it with the support material, solid material. But there are chances, right? The chances while you are doing that, it can lead to greater conformational change. Attach the bio, the enzyme structure may change bio. Enzyme 3D conformation may change bio, right? That's why enzyme efficiency enzyme it will be attached to the support material, but the efficiency of enzyme will be greatly reduced, right? So there can be extra, right, unnecessary chemical reactions chances right? So actually what you are trying, so your intention is to attach or non-essential amino acid residue with the matrix, right? अब के हुन सक्छ भन्दाखेरि similar kind of amino acid is also present in the active site. So actually active site को amino acid residue चाहिँ it will get attached with the solid support. If that happens, then the enzyme is not going to work. Enzyme catalytic efficiency will be greatly reduced. So what are some of the methods of immobilization? Some of the methods are diazotropin. This is the chemical reaction. So diazotropin reaction structure, right? This is the bonding between amino group of the support and the tyrosine or the histidine group of the enzyme है त यहाँ त्यस्तो किसिमको रियाक्सन्सहरु सक्छ है त यो यन यस्तो किसिमको बन्डिङहरु बन्न सक्छ है त यस्तो यन ट्रिपल बन्ड बन्न सक्छ है त यस्तो किसिमको है त यो चाहिँ मटेरियलको दिस इज द मटेरियलको हुन सक्छ यो चाहिँ हाम्रो एमिनो ग्रुपतिरको हुन सक्छ यस्तो किसिमको chemical bonding हुन सक्छ है त यसलाई चाहिँ we call it diazotization reaction or diazotization reactions हरु भन्छ And we have our peptide bond. Peptide bond हरु पनि बन्न सक्छ peptide bond चाहिँ कि amine group र carboxyl group बीचमा पनि peptide bond बन्न सक्छ or different functional group हरुबाट पनि बन्न सक्छ polyfunctional reagents हरु can use polyfunctional bifunctional or multifunctional reagents हरु that can form covalent bond with the amino group of the support and the amino group of the enzyme. So different किसिमको chemical bonding चाहिँ can occur. So what are the advantages of covalent bonding? So strong linkage of enzyme to support covalent linkage that is the most characteristic feature नै हो covalent bond एकदम यसको high activation energy हुन्छ so it is very difficult to break so no leakage or deabsorption problem सुरुमा जुन हामीले पढेको थियो entrapment र deabsorption method adsorption method मा जुन problem थियो leakage र deabsorption को that problem is more or less solved. So it is comparatively simple, right? Not very simple, but sometimes it can be quite simple. And there are a variety of support of different functional group available. And wide applicability. What is the major problem? The major problem is that the chemical modification of the enzyme can occur, leading to the loss of function of the enzyme. So chemical modification of enzyme leading to the loss of functional conformation of the enzyme inactivation by changes in the conformation when undergoes reaction at the active site. These can be overcome through immobilization in the presence of enzyme substrate or a competitive inhibitor. So यस्तो भइरहेको छ भने के हुन्छ भन्दाखेरि the enzyme को active site that is being used, right? So substrate छ भने enzyme enzyme को active site will be involved with the substrate or competitive inhibitor. The कसको चाहिँ बढी affinity छ, it depends upon that km value कसको lower छ, right? Substrate को or the competitive inhibitor को. If that's happening, it will be used up, then you can bind with the unnecessary number non-essential amino acid residue in polypeptide folding matcha that can be employed. So the next is the cross-linking. Right? The cross-linking so it involves formation of a number of covalent bonds between the enzyme and the matrix using bi or multivalent reagents. Right? Butyraldehyde, aldehyde, butyraldehyde, glycol, diisocyanates, hexamethane, diisocyanate, toluene, diisocyanate, right? Different functional groups are what you can attach. So similarly, generally amino group of the lysine, right, or the sulfur hydrol group of the cysteine, phenol OH group of the tyrosine, or the imidazole group of the histidine, they can be used for enzyme binding under mild condition. So the main advantage is this is the simplicity. There is simplicity, however, modification there can be enzyme ko conformational changes currently it can be enzyme ki non-functional probability or enzyme ko activity say reduced say probability is there higher. So further, this method of the enzyme immobilization suffer limitation caused by diffusion. And we have our ionic binding. Our ionic binding ma ke huncha? The enzyme molecules, it is charged, it is attached to the charged matrix. So here the higher surface charge density of the matrix, the greater will be the amount of enzyme being bound to the matrix. So higher the surface charge density of the matrix, higher the binding. Sometimes in addition to ionic binding, ionic interaction, enzymes are also physically adsorbed to the matrix. So there can be this either physical adsorption body, it can occur simultaneously. The method of enzyme immobilization is similar to that discussed in the section of the physical adsorption. Chemical binding via the ionic interaction during immobilization depends upon pH of the solution, concentration of the enzyme, and temperature. Right. So this is something we have studied many times. Right? These non-covalent interaction will depend upon the pH, it will depend upon the concentration of enzyme, it will depend upon the temperature. So commonly used matrices are polysaccharide derivatives, right? Diethyl amino ethyl cellulose, dextran, carboxymethyl cellulose, chitosan, synthetic polymers or polystyrene derivatives, polyethylene, vinyl alcohol, inorganic materials, amberlite, alumina, silicate, bentonite, sapiolite, and silica gel. So this method of immobilization leads to minimum change in the enzyme conformation. Enzyme conformation might minimum change compared to cross-linking and covalent modifications. However, special attention is given to maintain the accurate ionic strength and the pH of the solution in which the immobilized enzyme undergoes catalysis, since there is increased chance of enzyme detachment and the matrix under suboptimal condition. So a major attention major attention you have to give to in maintaining the ionic strength and the pH of the solution, right? If you can manage these two parameters variables, then the ionic binding can occur to the maximum. The enzyme it will get retained in the matrix. So the next is the conjugation by affinity ligands, right? So these enzymes can be attached to the matrix using specific ligands, that is by using histidine tags on the enzyme to measure containing matrix, right? So similarly, lectin containing domains to carbohydrate moieties present in the matrix, or sometimes substrate mimicking chemical compounds that are also used as ligands, right? These are attached by the help of affinity ligands. We have already studied about histidine tag. Histidine tag already studied how histidine tag is a nickel ion affinity right? Lectin containing domains they have these strong attraction towards this carbohydrate moieties है त एकदमै एकदमै यो लेक्टिन्सहरु भनेको चाहिँ एकदम कार्बोहाइड्रेट सँग चाहिँ बाइन्ड गर्ने चाहिँ यो चाहिँ एकदमै लेक्टिन्स प्रोटिन्सहरुले लेक्टिन भन्ने प्रोटिनहरुले चाहिँ दे एकदमै एफिनिटी हुन्छ कार्बोहाइड्रेट बाइन्डिङ सँग है त दे आर मेजर है त एकदमै यो मेजर क्लास अफ प्रोटिन्सहरु है त यिनीसँग दे आर दिस स्पेसिफिक डोमेन लेक्टिन प्रोटिनहरुमा and they can get attached to the carbohydrate moieties चाहिँ एकदमै यसको अफिनिटी हुन्छ And in some cases, ligands are naturally present on the enzymes, while in other cases, they are attached artificially by fusing nucleotide sequence corresponding to the tag with the DNA encoding polypeptide of a given enzyme. Recombinant DNA technology पनि युज गर्न सकिन्छ राइट कहिलेकाहीँ चाहिँ के हुन्छ अहिले चाहिँ नेचुरली पनि अकर हुन्छ दिज है त डोमेन्सहरु है त लिगेन्ड कन्टेनिङ लिगेन्ड अट्याच्ड हुने साइट्सहरु राइट अर यु क्यान एड जस्तै लेक्टिन्सहरुमा के हुन्छ कार्बोहाइड्रेट बाइन्डिङ डोमेन्सहरु दे आर अलरेडी प्रेजेन्ट पहिले नै प्रेजेन्ट हुन्छ लेक्टिन भन्ने प्रोटिनहरुमा अर यु क्यान एड राइट हिस्टिडिन nucleotides or histidine residue you can add with the help of recombinant DNA technology. This method of immobilization leads to minimal changes in the conformation of the enzyme with high stability and catalytic efficiency of the immobilized enzyme due to non-involvement of active site residues and higher immobilizing efficiency due to the presence of high densities of ligand on the matrix. So this method is very efficient. Right? And this method is not only useful for enzyme immobilization but also for several proteins including antibodies, cytokinins, and streptavidins. Right? Different antibodies हरुको लागि, cytokinins हरुको लागि र streptavidins हरुको लागि पनि these are used. Right? And enzyme immobilization by this method have found various application in biotechnology, diagnostics, and medicine. This method, the basis of this method has been used in animal cell culture by attachment of various types of mammalian cells to different matrices containing variety of peptides, growth factors, cytokinins to specific domain present on the cell as well as the activation. So, one of the most commonly used techniques are So what are some of the application of enzyme immobilization? Industrial applications, right? There is these biomedical applications, right? So drug delivery, it can be used. The enzyme immobilization can be overcome inborn error of metabolism or disorder by supplying the immobilized enzyme. So immobilized techniques are effectively used in drug delivery systems, especially to oncogenic sites. Right, cancerous sites or it can be sent. Food industry, right, pectinase and cellulase, if it's good, if they can be immobilized on a certain suitable carriers. Right, research right, and similarly, production of the biodiesels, wastewater management company, textile industry company, for detergent industry company, the heavy applications are so enzyme, if you don't want, if it's quite expensive to immobilize the enzyme, what you can do is immobilize the whole cell. Whole cell immobilize right? Immobilization of the whole cell is an alternative to enzyme immobilization, and it is well-developed method for utilization of enzyme from microbes. Right? Alternative, right? The alternative to enzyme immobilization. Enzyme immobilization can be quite costly. Right, so it's quite difficult and tedious. Efficiency is lower, right? That's why what you can do is that immobilize the whole cells, right? And concept here, the cells they house the enzymes, right? Actually, काम गर्ने त एन्जाइमले नै हो राइट कुनै इन्डस्ट्रियल बायो ट्रान्सफर्मेसन गर्नुपर्ने छ भने काम गर्ने त एन्जाइमले नै हो बट अब हाउ यु डेलिभर द्याट एन्जाइम है त होलसेललाई इमोबिलाइज गरेर कि द्याट पर्टिकुलर एन्जाइमलाई नै इमोबिलाइज गरेर So immobilization of the whole cells becomes particularly effective when the individual enzymes become inactive during the direct immobilization or the isolation and the purification enzyme is not cost effective, right? So enzyme immobilization for after there are hurdles and restrictions also. Economic hurdles uh, and there are these hurdles to purify and isolate and to immobilize. So the greatest advantage of wholesale immobilization is that here the enzymes will be active and stable for the long period of time since they are in their own natural environment. So since the enzymes they are actually entrapped inside the cells, these cells भित्रै भएको कारणले चाहिँ right. So this is also quite. It also offers many great advantages. It would be great if you could immobilize the enzyme itself, right? If we could immobilize the enzymes and they would retain, we could retain their maximum enzyme activity. But if you cannot, even wholesale immobilization, it offers some great advantages. So use of immobilized cells for fermentation is a very old practice. Bacteria or cells are immobilized by absorbing heat to the wood chip, and this practice in many parts for different types of fermentations. So what are the advantages of cell immobilization? Multiple enzymes can be introduced to a single step. Extraction and purification of the enzymes are not required. Enzymes are stable for the long time. Native conformation of the enzyme is best maintained. Cell organelles like mitochondria and chloroplasts can also be immobilized and it is quite cost effective. So what are the disadvantages? Concentration of the enzyme will be less, right? Because there are also other unwanted enzymes, right, in the cell itself, and production of unwanted enzyme and unwanted byproducts and products, and modification of end products by other enzymes produced by the cell enzymes. Okay, right. This is how we end, right? As a discussion, actually we end. So, do you have any queries about enzyme immobilization? Okay, sir. You probably been participating with you, right? पहिले पनि पढ्नु भएको थियो र पहिले र आजमा के छ डिड यु लर्न एनीथिङ न्यू
त्यसमा इन्ट्र्यापमेन्ट अनि इन्क्याप्सुलेसन पनि थियो तर यसमा हिस्ट्री नयाँ थियो एउटा हिस्ट्री पढेको थिएनौँ हामीले प्लस क्रस लिङ्किङहरू आयोनिक बन्डहरू पनि अहिले नयाँ भयो
Okay, these are some of the methods. Right? This is one of the most exciting area of research right now. So applications in wide, diverse sectors from industrial production, right, biomedical application, right, diagnostic therapeutics, food industry, right. So repeatedly, अब setup जुन chemical transformations right, हामीले जे पनि use गर्नेछौँ नि right, every kind of material we consume, right, every kind of foodstuffs or materials we use or we consume, they are the result of certain kind of transformations, right, the chemical transformations are bhayiरहेको. Now we want to carry out those transformations using enzymes, right, because we want these transformations to become more bio-friendly, environmental friendly. That's why enzymes there has been lots and lots of interest. So and immediate impact केमा देखिन्छ भन्दाखेरि enzyme immobilization चाहिँ एकदमै इम्पोर्टेन्ट छ फेरि किन भन्दाखेरि यसलाई चाहिँ बायो ट्रान्सफर्मेसन बायोट्रान्सफर्मेसनलाई चाहिँ म्यासिभ स्केलमा गर्नु छ भने these enzymes they have to be immobilized. Right? That's why we studied about enzyme immobilization. Right? We will study further what are the different kinds of bioreactors बारेमा पनि पढ्छौँ where these immobilized enzymes can be used. Right? And यो similar lectures हुन्छ है अरू क्लासको lectureसँग म्याच हुन्छ. That's why I'll go quickly. म quickly जान्छु. So as always, the slides are on the Dropbox. You can please access them. You should access them, and that will be easy. Okay. Okay, questions or somebody you can stay, or else the class is over. Okay, questions or somebody you can raise them now. Else you can ask them tomorrow. Class over. We should stop recording.